안녕하세요, 여러분. This lecture is about comparing different pollution investigation methods and results when examining marine litter in various environments and conditions. The locations to be investigated vary depending on whether it is a beach, seawater, sea bottom sediment, organisms, and etc. And there are varying differences depending on the size of the marine litter being examined as well. Many of you may have been interested in or have a future plan of microplastic study, so today I will compare and explain these different methods as easily as possible. The definition of marine plastic sizes has changed in the past decade, over which time growing scientific, public, and political interest has focused on the smaller size fractions. Today, research into the formation, features, future fragmentation, chemical interactions, environmental fate, and potential impacts of microplastics is increasingly abundant. The term microliter was used in 2003 by Gregory and Andrade to describe the fine fraction of marine plastic litter with sizes of 67 to 500 micrometer. Similarly, mesoliter, macroliter, and megaliter were defined in the same paper. In 2004, the term microplastics was for the first time popularized in Richard Thompson et al.'s paper to describe truly microscopic plastic fragments with typical diameters down to 20 micrometers. While this paper described the accumulation of microplastics in the seas around the United Kingdom, it didn't define them. In 2008, experts attending a meeting hosted by NOAA United States proposed a working definition in which microplastics are all plastic particles smaller than 5 millimeters in diameter, which has become the most frequently used definition. So far, marine litter sizes are mainly classified into macro, meso, and microplastics. Examining the lower bound of microplastics will depend on detection capability, equipment, and analysis technology. It is extremely rare to examine nanoplastics smaller than 1 micrometer. While marine litter ranging from mega to meso is visible with the naked eye, microplastics are only visible if they are larger than 1 millimeter in size. Microplastics smaller than 1 millimeter are difficult to detect without the use of special equipment. In 2019, GESAMP, Joint Group of Experts on the Scientific Aspects of Marine Environmental Protection, especially a group of marine litter experts, published a report on monitoring and assessment of plastic litter in the ocean. This report comprehensively compares and evaluates the survey methods for shoreline, seawater, seafloor, and marine biota. For a detailed look at the report, visit www.gesamp.org. After the review, the experts provide recommendation. May I introduce a little bit about it? The intended users of guidelines for harmonized methods are UN sponsoring agencies, other sponsoring organizations, intergovernmental organizations and conventions, regional seas organizations, and also civil society and private company. This table is a compilation of the contents of this report, showing a list of environmental compartments and litter size categories. summarizes the resource requirements like personnel and equipment for each combination, and it provides examples of typical policy questions that government agencies may be facing. So number one, the first column means more feasible, and number 7 means less feasible based on resource sampling and processing requirements. The red color, the cost increases from left to right. Right part of the table is about examples of policy concerns. It includes distribution and abundance of plastic litter, source identification, and impacts on tourism, seafood safety, human health, and injuries, fisheries and aquaculture, etc. SL stands for shoreline, and SF stands for seafloor. BE stands for beach, and V stands for invertebrates. So the lower the feasibility ranking, the easier it is to investigate. R stands for recommended. So the more R's in green, the easier it is to investigate. And as you can see, policy concerns is relatively high in distribution and abundance and biodiversity. Going forward, I will be talking more about the details on the method, equipment, and data result. First, this slide is about survey methods on a beach. Micro-size marine litter on beaches are most accessible and easiest to investigate. The smaller the size from meso to micro, the harder it gets to investigate. Let me give you more details. It is common practice to determine the length or area of a beach before counting and measuring the weight of marine macrolitter distributed within the area. Usually, this marine litter can be collected by hand and then recorded directly onto the data card. Using a scale for weight, the number of units per area or unit length, and the weight can also be determined. Then the result is divided according to material or cause of generation. As shown in the picture, you can investigate with very simple equipment such as 100-meter tape measure, gloves, survey cards, and reference scale for size, etc. When investigating macromarine litter, you will most likely find out about abundance. You will see how many pieces of litter there are per unit length or unit area, and also see how many kilograms of litter you've collected. Then you can distinguish whether the material is plastic or metal, glass, rubber, or wood. You can also create a geographical distribution map like this. The good thing about macro litter research is that you can get a lot of information about the cause or source of the occurrence. You can tell whether it comes from shoreline activities or from smoking. By identifying the top 10 items, you can see which litter is the most abundant and what should be reduced first. To study meso or micro-size litter, you usually set up quadrates on the beach and sieve the sand to collect meso or microplastic samples. The mesh size of the sieve determines the size of the study target. Further analysis is difficult to conduct out in the field, so we usually transfer the samples back to the laboratory. The samples transferred to the laboratory are classified as natural or artificial litter. As for plastic litter, they are further categorized by shape, such as pellets, fragments, sphere, or film, and by the types of polymers such as polypropylene, polyethylene, or polystyrene. As the plastic size becomes smaller from right to left, it becomes increasingly more complex and difficult to identify and measure. Figuring out how to separate the samples, removing organic materials, filtering, and distinguishing their shapes and types of polymer can be difficult tasks. If we look at the equipment required for sampling beach sand, they are materials that are simple and easy to purchase. That is particularly the case if you are collecting major-sized items. However, for micro-sized items, it is important to keep the laboratory clean. Additional microplastics can contaminate samples from the air of the laboratory or even during microscopic observations. Samples can also get contaminated from fiber microplastics if the lab coat is not cotton. It's not easy to separate microplastics from the specimen. If it is smaller than 1 millimeter, it is not easy to separate it from pieces of wood, seashells, or sand grains. In the case of smaller microplastics, it is impossible to see with the naked eye. Microplastics should be separated in a very clean laboratory with thoroughly sealed areas to prevent contamination. Next, you can distinguish whether the plastic type is fragment, fiber, sphere, or film. If the size falls below 300 micrometers, it may be difficult to tell whether it is fiber or not. Then you use a device such as FTIL to determine if it is a polypropylene or polyethylene or polystyrene. You should determine one by one. It takes very long time. The smaller the size, the greater the likelihood of errors in the analysis process, which often makes it difficult to trust the analysis result. So quality assurance and quality control are very important and necessary. The results are expressed in abundance or density. Measuring the individual size of microplastics can help to make a histogram chart. Like here, we can draw a pie chart according to the types of plastic or polymer to determine what forms or composition are high. In the case of observing marine litter in seawater, you can observe large litter from a ship and count them. In the case of microplastics, samples are collected using a Mento troll, or samples are collected by taking a certain volume of seawater and studying the microplastics contained in it. Mento trolls generally have mesh sizes of 330 micrometer, and this is also the most common method of collecting microplastics in seawater. Whether you are using a method or bulk sampling of floating microplastics, the analysis process for both methods are almost the same. However, it is important to note that bulk sampling seawater can result in higher concentration of microplastic litter because they can collect items that are much smaller than 330 micrometers. So it is important to note that results may vary significantly depending on the method and the size. Marine litter on the sea bottom, marine macro litter on the sea bottom is more difficult to investigate. First of all, it is difficult to approach. Therefore, sampling is expensive and inaccurate. When investigating large-scale marine litter, you can use the collecting gear as you see in the picture, or obtain images indirectly using a side-scan sonar. In the case of microplastic, you can collect and analyze deposits with a grab sampler or a box cutter by a certain volume. There is also a way to pull the trowlet along the ground or to record marine litter by directly observing in underwater by a scuba diver. Microplastics in sediments taken from the bottom of the ocean follow this process, which is almost similar to the microplastic research process in seawater or beach sands. It is also very important to study how much marine life is exposed to microplastics and how they are affected by it. So it is important to choose which target life is selected and how to remove organic materials so that we are only left with microplastics. Invertebrates, fish, and seabirds or sea turtles have been used as indicator species for different research objectives. Overall, field studies of marine litter are conducted in various media, sizes, methods, and units. Therefore, it is very important to present the data so that it can be compared with each other when it is released. In some cases, the number or weight per square meter or cubic meter is presented, and in other cases, it is presented in per kilogram. Presenting or converting results into units that can be compared to other studies can increase the utilization of the findings. I would like to emphasize this. The study of marine litter in various sizes have been conducted in various ways in beach sands, seawater, and seafloor, and marine biota. What I want to emphasize again here is that plastic breaks into small microplastics and does not melt away. The smaller the size, the harder it becomes to understand the unique information it has and the cause of its occurrence. To solve this problem, research in large sizes, such as meso or macro sizes, should be active. Thank you very much for your attention.